Meiosis, sexual reproduction, and genetic diversity. So in this lesson, we're going to review the stages of meiosis and how they contribute to sexual reproduction and genetic diversity. We are going to recognize that sexual reproduction leads to genetic diversity, and we are going to identify the need for genetic diversity in the survival of species. So first, we're going to go back and we're going to talk about meiosis. So meiosis is the type of cell division in sexually reproducing organisms that reduces the number of chromosomes in half. It is important to talk about the reducing the number of chromosomes in half because at first we start out with 46, but we want to end up with 23. We will discuss more with that when we start talking about meiosis itself. In the end of meiosis 2, 4 gamete cells are produced, and there are 2 stages of meiosis, meiosis 1 and meiosis 2. We will review both of them and talk about the key parts of meiosis that affect sexual reproduction and genetic diversity. So here we have meiosis 1. We see in prophase 1 where the homologous chromosomes pair up. This is also where crossing over or recombination happens. These paired chromosomes are called bivalents. 2 chromosomes and 4 chromatids with 1 chromosome coming from each parent. So this is where I want to highlight this image here. This is where we see these bivalents. We see the 2 chromosomes from each parent, all containing 2 chromatids. So we'll say, these could be from the female, these could be from the male. Prometaphase 1 begins when a kinetochore forms per chromosome and it attaches to the spindle fibers, which will be what end up pulling these chromosomes apart to either side of the nucleus. Once metaphase is fully engaged, the homologous chromosomes are lining up along the metaphase plate. They are getting ready to be pulled apart. That is when we hit anaphase 1, where the chiasmata separate and move to the separate poles. We see that here. They've been, the kinetochore is attached to the spindle fiber, and they are being pulled apart. At this point, we move on to telophase 1. Telophase 1, the cell may start meiosis 2, or the nuclear membrane could reform like we see in this picture here. If it does reform, the cell will continue on through cytokinesis and become 2 complete daughter cells. So if this went through cytokinesis, this would split down the middle and we would have 2 complete daughter cells. But in order to keep this, we're going to keep moving on, and we are going to start talking about meiosis 2. So when we hit meiosis 2, again, we start with prophase. Prophase 2, the sister chromatids begin to condense and a new spindle begins to form. So we're seeing that here. These chromosomes are condensing again. And at prometaphase 2, the kinetochores are engaged, and the sister chromatids and the nuclear engage on the kinetochores on the sister chromatids are engaged. And the nuclear envelope disappears because it's getting ready to go to metaphase 2, where the sister chromatids line up along the metaphase plate. This is just like we saw in meiosis 1. This is just like we see in mitosis. So again, we've talked before, mitosis and meiosis are very similar, but there are some very significant differences that we are running across as we talk about this. So after metaphase 2, we hit anaphase 2. The sister chromatids are pulled apart and separated. So we would see this here, where if this fully happened, these chromatids would be pulled apart, and we would end up with telophase 2, where the chromosomes arrive at the poles and decondense. They relax. They aren't these nice put together structures. Once this happens and the cleavage furrow divides, that would be a little cut here. This is when we end up with 4 haploid cells. Haploid meaning 1, because as we see in each of these cells, there is a single chromatid. Where we started before, we had homologous chromosomes, pairs, now we have single chromatids. So it has gone through meiosis 1 and meiosis 2. So, when we look at this image and we see these chromatids, we see that there are different colors. As we started from the beginning, we can see these are two. We could think of these as the male and the female parts. And as we know from prophase 1 in meiosis, this is when recombination, this is when crossing over occurs, and we can see the final comings of that. So let's talk more about crossing over. Let's talk about really what it means. So genetic diversity and genomic diversity are important in the survival of the species. This happens through the process of meiosis due to chromosomal recombination and independent assortment. The law of independent assortment states that the random orientation of homologous chromosome pairs during metaphase 1 allows the production of gametes with different assortments of homologous chromosomes. When Gregor Mendel was studying his pea plants, this is one of the laws he came up with because he was seeing these things in his pea plants. He was seeing how the gametes were different than the parents. And he figured out that crossing over, also referred to as recombination, this was happening. So crossing over, also referred to as recombination, takes place during prophase 1 of meiosis. When the homologous chromosomes come together to form a tetrad, meaning 4, the arms of the chromatids swap at random. So if we think back to the previous image, in meiosis 1, we had the homologous chromosome pairs, and we could see where the arms were swapped at random. Now, when this swapping happens, it's important because this creates more possibilities for genetic variation in the gametes. This is what is part of what makes these gametes unique from the parents, is this crossing over and recombination. Now, speaking of gametes and parents, the next step that we need to talk about in order to better understand this is sexual reproduction itself. According to Britannica, the definition of sexual reproduction is the production of new organisms by the combination of genetic information of two individuals of different sexes. So we have sexual reproduction and we have asexual reproduction. Sexual reproduction, we have to have the two different sexes, and that is what creates the genetic variation. When we think about asexual reproduction, this is when the offspring identically matches the parent. There is no genetic diversity. And that works for some organisms, but for a majority of organisms, it does not. So in sexual reproduction, for sexual reproduction to occur, a sperm and an egg, also known as an oocyte, must come together. The sperm coming from the male, the oocyte coming from the female. The sperm is a haploid cell, as is the oocyte. Now, when they join together, when fertilization actually happens, a new diploid cell forms. This is called a zygote. From the zygote comes the next steps in development of the fetus and the offspring. When this happens, when we see it go from fertilization to an actual offspring being produced, is when we see the DNA, the genes recombining, because we can see some of the parental, and we can see, some of the male and some of the female in the organism, in the offspring, and we can see that genetic recombination happening. So the zygote is a combination of the DNA from the sperm and the egg, and that genetic recombination comes along during, like I said again, prophase 1 in meiosis. So we understand how sexual reproduction works versus asexual, and we understand how important it is to have this happen because of genetic diversity. So let's dive into genetic diversity. Genetic diversity is the biological variation that occurs within a species. This allows species to adapt and survive. That's the important thing, is that adaptation and that survival. Because if a species is not able to adapt and survive, they can die out. So, it may be part of a population. If that part of a population cannot adapt and survive, they will die out. If enough of that happens throughout a population, it's going to affect the overall species. And by even just losing a single species in an ecosystem can have drastic effects to the ecosystem overall. So thinking of that and thinking about adaptation and survival, we go back to the fact that genetic diversity starts at the individual level. Each, the same gene may occur in different variants, alleles. This equals genetic diversity. Individuals carrying 2 copies of every gene, 1 from each parent. So as the males and females sexually reproduce, the offspring is receiving genes from each parent. Some genes are stronger than others, and these are going to be the genes that we see expressed. At the population level, more individuals are reproducing and more genetic recombination is happening. So these strong traits, these strong genes are being passed on, and that is helping these species to adapt and survive. This will allow for favorable traits to pass on and allow the species to continue thriving, because just losing a single species in an ecosystem, while it may seem minor, can actually have some pretty devastating effects on the ecosystem, and that is something that conservationists are looking at and thinking about. So let's talk about some consequences of loss of genetic diversity. So a prime example of the consequences of loss of genetic diversity is the black bear, Ursus americanus. These animal populations have declined sharply in population size and range in North America. A study was done in Alabama of the black bears that live there. The findings showed low to moderate genetic diversity when compared to populations that surrounded them, populations of other species, really. A concern is that as the population sizes decrease, the amount of genetic drift will decrease as well. This will lead to a reduction of genetic variation. Without that genetic variation, the species is really going to suffer. It's not going to thrive. There's every chance that it could go extinct. So without this genetic drift, this will lead to a reduction in genetic variation. And breeding in small populations will also lead to a loss of genetic diversity and will threaten the species. When you have a lot of inbreeding, you have a lot of sharing of the same genes over and over again. And this is when a lot of these unwanted and not strong genes, I should say, are going to start presenting themselves. And that is when you see a lot of issues, a lot of problems arise. A lot of birth defects, a lot of times the animals aren't strong enough and will not continue to thrive and survive. We've gone over a lot today, and I'm hoping that, through everything we've learned a lot. We've really learned about how meiosis, sexual reproduction, and genetic diversity are incredibly important, not just to humans, but to other organisms in order to keep their survival, keep them going. So how do meiosis, sexual reproduction, and genetic diversity go together? During prophase 1 of meiosis, there will be a recombination of genes. This is where we see the random arms of the homologous chromosomes swapping. This is where we see some genetic diversity. This recombination allows for the gametes to have traits from both the male and the female. In order for a zygote to be produced, a sperm and oocyte must combine. This is sexual reproduction versus asexual reproduction. Genes from the male and the female will be passed on to the offspring, as again, the zygote will have genes from both parents. The genetic diversity that comes from sexual reproduction is crucial in the survival and fitness of a species. We saw this with the black bears, and we saw how that small population size we're going to see less genetic diversity. We're going to see less genetic drift, and we are going to see a decline in the fitness of that population of bears. If a population is small or isolated, genetic drift will be limited and genetic diversity of the population will suffer. This could lead to problems in the population or in the loss of the species in that population. Like I said before, the loss of a species in a population might sound minor, but in the grand scheme of things, it will affect an ecosystem overall. I hope that through this lesson, we have learned some new things, we have gone over some things we have learned before, and that you now know more about how meiosis, sexual reproduction, and genetic diversity go together and are important for the survival of a species.